that I have said this, I think we have a better idea of what light is and how we can manipulate it, and also how the image that we are recording is formed. So now I want to put again this key concept of light microscopy that I spoke about before, just so that you understand maybe what I meant. The image of a fluorescent object is the result of refraction of the emitted light, the light that comes from our sample, that goes through lots of media with different refractive indices. That can be our sample, which is heterogeneous. It can be the cover slip, glass cover slip. It is also the immersion medium and the lenses. All this will affect the image of our fluorescent object through refraction. But it also depends, the image of our object also depends on how much light finds its way into the objective. So it depends on the numerical aperture of the objective. And also it depends on the way light diffracts at the edges of the inner edge of the objective. And that is also the numerical aperture. So this one is how much light, so that's the brightness, and also the way it diffracts. And so it depends on the 3D interference pattern that we record at the image plane. So this means that without diffraction and interference, there is no image. Okay, what we are recording is diffraction and interference. Without that, we don't have an image. That's it. So we have nothing to image. So therefore, anything that affects the interference pattern, so the point spread function, for example, refraction, will affect the image. That's where your role comes in. You would like to avoid that you affect the point spread function. This is the reason why I said that the image of an object is never and cannot be like the object, because what we're imaging is the point spread function. We're imaging the way the light interferes or interacts with the sample and the microscope elements. So it cannot be the same. Therefore, for you, preparing a good microscopy sample is mostly about minimizing refraction. And that will have an effect both on the excitation and the emission lights. And you do not want your sample to act like a kaleidoscope, basically. So this is why we talk about refraction index a lot, because it has an effect on the point spread function, and therefore it has an effect on your image, okay? So this is what is important when you prepare a good microscopy sample. We will see what a good microscopy sample is later. So the optical resolution that delivers light is limited by the light wavelengths, and it's also limited by the numerical aperture of the objective and also the refractive index of the immersion medium. So these are the things that will affect the optical resolution. And then we have an extra layer, which we will talk not next week, but the week after, which is the digital resolution. And the digital resolution is the way we actually record the image. So that is the pixel size. So that also adds to our resolution in the image. So the bad news for you is that to fully describe a sample with very fine details, for example, if you look at vesicles, whatever sample you have, it will have edges, right? You have the edge of the nucleus, you have the edge of the cell. All these edges are fine details. So all samples have very, very fine details. And to fully describe such a sample, we would require infinite resolution. That's not good because we do not have infinite resolution. So it is not possible, that's what I said, it cannot be like the object. It is not possible to have an image that fully represents the sample. But the good news is that most scientific questions do not require you to fully describe the sample. And most of the time, you do not even need the maximum resolution of the imaging system. So you can see in here the very great importance of how you formulate your scientific question within the limits of microscopy, which is that we will never be able to describe fully the sample. It will never be exactly the same. So please design your sample to refract as little as possible. What does that mean? Place your sample on the cover slip if you can, not on the slide. If you cannot put it on the slide, for example, because you are boiling your sample for oxygen retrieval, then use a minimum amount of mounting medium, okay? And then match the refractive indices of the objective and the sample and the mounting medium, all this, to prevent more refractions, prevent the sample from looking like a kaleidoscope. And again, we will talk about this later in the course. You will only notice the difference between a good and a bad sample if you compare them side by side. So if I see your image and I say your image would be better if you had your sample on the cover slip, you're not going to believe me. But if I show you, then you will see it. So this is the same sample. It's the same objective, the same settings, acquisition settings, same microscope, everything. And in both cases, we were acquiring at the best focus possible. And what happens? Do you think we have the same resolution in this image and in that image? Which one do you think is easier to segment? Because in this one, I can assure you we're not out of focus here. This is the best focus we can get. So this is what happens when we design a sample that creates refraction. But if you have this, you cannot really guess how much better it would be if you had your sample on the cover slip. So this one is on the slide and this one is on the cover slip. Okay, so this is easier to segment. We have more stable intensity, so less fluctuation, and we have a higher sample-to-sample reproducibility with this. Okay. So improve contrast. If you improve the contrast here, you actually improve the usable resolution in this case. So it's easier to segment. So keep in mind also the limitations of a microscope. We cannot have infinite resolution, and if you need to have your scientific question designed for this. If your scientific question requires you to work at the limits of the resolution of the microscope, you should change technique or you should change scientific question. So working at the limit of the resolution of the microscope is extremely, extremely risky. So this is my opinion. Okay. And I just want to say that there's a very, very nice demonstration of all this stuff that I've been telling you about diffraction and interference in this little video that you're welcome to watch later. So I would like to know if there are any reflections on this, what I've been telling you so far.
I've got a question.
Yes.
So you said to use as little mounting medium as possible. How do you know kind of the compensation between using as little as possible and then avoiding that the sample dries out?
Yes, absolutely. We need to have enough mounting medium to cover the sample completely. But what we do not want is that the mounting medium lifts the cover slip away from the sample. So we would like to have just enough mounting medium so that the cover slip is against the sample. Right? We do not want to have like a big sandwich of a big layer of mounting medium between the objective and the sample. That's what we want to avoid, yeah? So I know that we've tried a lot at our facility to ask people to put their tissue section on this cover slip instead of the slide. And we'll talk about all this later in the sample preparation lecture and day. But when we have antigen retrieval, it's difficult because it detaches, unfortunately. So then if you cannot avoid putting, and this is the reason why when you use like monolayers, it's very easy because you just seed them on glass. You can seed them, you should seed them on the cover slip, not on the glass slide. I think there was someone who was seeding their cells on the glass slide. It's much better to seed them on the cover slip. And then it means that when you're imaging, the mounting medium is not between the cover slip and the sample. But if you can't avoid it, just put as little as possible. So what you can do, and Gabriela will explain all this to you, but you can do a test and put, okay, what if I put 3 microliters? What if I put 4 microliters? You can do a test before you actually mount your sample. And then you will know exactly how many microliters you need to use.
Thank you.
You're welcome.
I also have a question. You mentioned that to reduce the refraction in the sample as much as possible, one can match the refractive index of the objective sample and mounting medium. How do we know the refractive index of the sample?
Yes, that's a good question because it's mixed, right? So as you have seen, the light, when the light refracts, it's going to change direction, right? So if it changes direction for a very short time before it's imaged, then it's okay, you don't see so much effect. But if it travels like this for a very long time, then the effect is very big. So this is why we want to reduce the amount of mounting medium. It's the same thing in a sample. If you have a thin sample, it will have a little effect on how much the light travels. But if you have a thick sample, then the effect is very big. And this is one when you have a little bit thick. And when I say thick, people who image a monolayer of cell, they know that when you image the bottom of the cell, which is against the cover slip, or you image the top of the cell, you already see the effect of refraction. The top of the cell is blurrier than the bottom of the cell. It's always like this. And that is because of the refraction index mismatch. So if we have a sample that is even a little bit thicker, we're even talking about 10, 15 nanometers, microns, sorry, or like 20 macrons. When it's like this, then you need to do something which is called optical clearing, which we will talk about, and homogenize the refractive index. So you do some chemical treatment to make the refractive index of the sample homogeneous to avoid. So this is the way, but that we will talk about again later during the sample preparation day, how we can do that. Any more questions?
I have one question. So if I am using the water lens,
Yes.
should I submerge my sample also in PBS, but
Yes,
not?
absolutely. So water objectives are excellent samples to image live samples because so it depends again, of course, on the cells that you're imaging. If you're imaging cells that have a lot of fat, for example, adipocytes, then you're a little bit more in trouble. But very often, if we image cells that do not have a lot of fat, so the cytoplasm has the same refractive index, a little bit higher, but not very much higher than water. And then your cells are live, so you put them in a culture medium, and this has more or less the same refractive index also as water. And so in that case, the best objective for live samples is, because of that, is a water immersion sample. Yeah.
Okay, thank you.
Yeah. Anyone else?
Can I ask a question too, please?
Yes.
Yes. So, how much mounting medium should one usually add for it to be enough? Because what we usually do in the lab is to add maybe more mounting media than we need so that it covers the whole cover slip and also the whole sample to make sure that there are no bubbles in between the slide and the cover slip.
So I think Gabby will explain all this to you during the sample preparation day, but all these sort of things, they do make a difference. They absolutely do make a difference. So it is important to put as little as possible and not put a big blob, and certainly make no bubbles. So you will hear all this in the sample prep day. Yeah, and we have like even little videos and stuff for you to look at. You can even to practice.
Great. Thank you.
Yes. Good. So if you guys have no more questions, we're going to go now to the workshop Zoom to have a little quiz. Okay. See you guys there, and then we'll be back here at 11:25.